Okay, now I invite Rosanna Parlato. She's a senior scientist at the Department of Neurology, University of Heidelberg. She will talk about neuropathological function of the TANK1 binding kinase 1, TBK1, in mouse models of Huntington disease, and she has a poster labeled A36.
So I would also like to thank the organizer for giving me the opportunity to present the initial results on our project entitled Neuropathological Function of the TBK1 in mouse models of Huntington disease. First of all, a few words about TBK1. This is an ubiquitously expressed serine threonine kinase regulated by multiple post-translational modifications, and it regulates many functions, among them the interferon type 1 and pro-inflammatory cytokine production and autophagic degradation of protein aggregates. In the context of autophagy, in particular, it is important to say that phosphorylation of TBK1 is important for its activation, and in this way it promotes the binding of adaptor proteins to target cargo. Adaptor proteins are, in this case, optineurin and P62. And in principle, it facilitates the formation of the phagophore by enhanced binding to LC32, and then in this way promotes also membrane elongation and autophagosome formation. So our group several years ago has actually identified many mutations in the TBK1 gene that cause familial ALS, amyotrophic lateral sclerosis, and frontotemporal dementia. And in particular, here there is in the C-terminal domain, in the C-terminal domain, there is the binding site of optineurin, and our group identified this missense mutation in which glutamate in position 696 is exchanged with lysine, and this is important for the optineurin binding. When I joined the group of Jochen Weishaupt 2 years ago, there was also this inspiring paper from the group of Ilan Lashuel showing that TBK1 phosphorylates mutant huntingtin and suppresses its aggregation and toxicity in Huntington disease models. And so then we decided after that to look a little bit more into the detail of the question if the loss of TBK1 optineurin binding would affect the accumulation of mutant huntingtin inclusion. And we took advantage from a mouse that we had with carrying the mutation that shows a deficit in autophagosomes and represented here by increased cytosolic inclusions that are positive for P62. And we crossed this mice with a ZQ model that, as you all know, show age-dependent Huntington disease-like phenotypes and progressive accumulation of mutant huntingtin inclusion. And here we show then that in comparison to the single ZQ mutant, the double mutant have an increase of extranuclear mutant huntingtin labeled here by the EM48 antibody. That is, well, we have only a trend for the intranuclear one. So we show that specific loss of TBK1 optineurin binding affects accumulation of mutant huntingtin inclusion in the mice. And we could also show first evidence that also phospho-TBK1 is downregulated in cortex in different stages, so at 6 months and 1 year, suggesting that this might be also having a pathological relevance in the context of the mice. And with this, I want to acknowledge the contribution of my colleague, in particular the two students indicated with the white arrow, Ibrahim and Yasmin, that have been working for the last year on this project. And of course, our lab chief, Jochen Weishaupt, to support always this project, and also a special thank to Patrick White, actually, who made us aware of this publication back then. So, and thank you very much for your attention.
Thanks, Rosanna, for the great talk. Any questions from the audience? Okay, meanwhile, I have just a curiosity about phospho-TBK1. So basically, you showed an increase. To the best of my knowledge, so phospho-TBK1 should be also very well localized in some cell compartments. So for example, in centrosomes or mitochondria based on the pathway that is activating. Yes. So do you have any?
Yeah, we are following the subcellular localization of phospho-TBK1 also in the Huntington mice. It's very difficult to do this in vivo since the antibodies are available are not really perfect, but we could show that there is even some phospho-TBK1 in the nucleus, for example. But that I cannot, we need additional analysis to confirm it was not quantified properly yet. And yes, phospho-TBK1 is here, as I probably briefly indicated, it can interact with mitochondria that are destined for degradation. So it has been also actually involved in other neurodegenerative diseases like Parkinson's disease. So there is some commonalities, and this also spread a little bit my question to Christian before, because we do have also some noxic data with these mice, and it would be nice maybe to share some of these changes with the available database with Huntington too. Yeah.
Okay, thanks. Any other question? Okay, so probably thanks again. We can move.